Hello everyone, welcome. Today we'll be reviewing Mendel's law and Punnett squares. Let's go ahead and get started. So first off, there are a few terms you need to be familiar with before we start practicing Punnett squares, like allele. An allele is a type of a gene. Each gene has two alleles. For example, if eye color is the gene type, then the color would be the allele. So here, brown eyes is one allele, and blue eyes is the other allele. Another example is if hair texture is the gene type, then curly hair is the allele and straight hair is the other allele. So remember, we have 23 chromosomes in our body. Let's zoom into one of these chromosomes and find the alleles in them. Remember, one chromosome is made of two chromatids. One is maternal, coming from the female, and the other is paternal, coming from the male. So here we will find our alleles. Both of these specific alleles code for one type of gene. That gene type is hair texture. This one codes for curly hair, and this one codes for straight hair. When we have two different alleles, this is called heterozygous. If both of these alleles coded for the same allele, in this case curly hair, then it would be called homozygous. Remember, organisms have two alleles for each gene, one from each parent as seen here in this example. These two alleles can either be dominant or recessive. Dominant is the trait that is expressed, and recessive is the trait that is not expressed. In other words, dominant is manifested and recessive is hidden. A gene is represented by a specific letter. In this case, we represent hair texture with the letter H. One of the alleles will be dominant and the other will be recessive. We represent the dominant allele with a capital H, and we represent the recessive allele with a lowercase h. So here we have a capital H representing the dominant allele or trait, and a lowercase h representing the recessive allele or trait. When we say the alleles are heterozygous, we already know that means the alleles are different. This is what it looks like, a capital letter and a lowercase letter together. And when we see homozygous, we know this means both alleles are the same. This is what homozygous looks like. Either we have two uppercase letters together, or we'll have two lowercase letters together. Let's say we're given the following information. Big H equals straight hair, and little h equals curly hair. When the dominant allele is present, it will mask the other allele. We can only have three possible allele pairs here. Big H, Big H, and little h. What outcome will we get with the combination of Big H, Big H? Will we get straight hair or will we get curly hair? We will get straight hair because the dominant allele is present. In other words, the big H is present. How about big H, little h? Again, we're going to get straight hair because again, the dominant allele is present. The big H is present. How about little h, little h? We will get curly hair because there is no dominant allele present to eclipse the recessive. The only way for a recessive gene to be expressed is if it is in this form, homozygous recessive. Because in this form, there is no dominant present to overshadow it. So the recessive allele can go ahead and shine. It can go ahead and manifest. Okay, so this girl has curly hair. What possible allele pairs could she have? Little h, correct. Only this pair will yield curly hair. How about this girl? She has straight hair. What possible allele pairs could she have? Yes, she could have big H, or she could have big H, little h. Both of these would yield straight hair. By the way, we're going to start referring to these allele pairs as genotypes. Genotypes is the letter combinations, and genotype refers to the genetic makeup. The genotype is not visible, as opposed to the phenotype. Phenotype is referring to the characteristic traits expressed by the genetic makeup. The phenotype is observable. For example, this girl's phenotype is big H, or big H, little h. Her phenotype is straight hair. Okay, so now that we know the basic terminology, let's look at some Punnett squares. We'll first look at a monohybrid Punnett square. We use these to examine only one trait. Parent 1 and 2 go on the sides like this. And remember, we're studying one trait or one gene. So one gene will have two alleles. So for each parent, we place those two alleles here and here. What's inside the boxes are the possible offspring outcomes. So each box represents 25%, and of course, it adds up to 100% as a whole. So let's look at our first practice problem. A homozygous cat without fur mates with a heterozygous cat with fur. Fur is a dominant trait. What is the possibility the offspring will have no fur? And what is the genotype ratio and the phenotype ratio? Okay, here's our Punnett square that we're slowly going to fill in. But first, notice one very important piece of information given. Fur is dominant. So by default, no fur is recessive. First off, let's find the variation of genotypes we can have. We can have big H, big H, little h, and little h. These allele variations or genotypes can provide some information for us. Like, we know because we have two capital H's that this is dominant and it will yield a phenotype with fur. We also know it's homozygous because it has two capital H's. For big H, little h, we know this is also going to give us a phenotype of fur because there's one dominant allele present. We also know this is heterozygous because there's a capital H and a lowercase h. For little h, we know it will give us a phenotype of no fur because we have two lowercase h's here representing no fur. There is no dominant allele here to mask the recessive allele. So the recessive allele here is able to manifest. We also know this is a homozygous because both letters are the same. They're both lowercase. So we're going to use this information to find out what genotype fits in the parents of these cats. The genotype of each parent will fit into these four areas here on the Punnett square. Let's work on parent number 1. Here it says we have a heterozygous cat with fur. We also know this cat parent is dominant because it tells us right here the fur is dominant trait. So we need to find a genotype that is heterozygous and will produce a phenotype with fur. Big H, little h fits that description perfectly. Big H, little h is a heterozygous genotype and its combination yields fur. In other words, it will give us a cat with fur. So we're going to put the genotype right here to represent the cat parent with fur. Next, let's work on the second cat parent. Here it says we have a homozygous cat without fur. So we need to look for a genotype that matches that description. Two genotypes are homozygous, but only one of these is going to yield a phenotype without fur. So we're going to put that genotype into the Punnett square to represent the cat parent without fur. Next, we're going to go ahead and fill in these boxes. So big H and little h gives us big H, little h. Little h and little h is going to give us little h, and so on. So remember here that a capital letter represents the dominant trait. And if there is a dominant trait present, it will mask the other allele. In other words, the dominant trait will be expressed. So here we can say 50% of the offspring will have fur because they have a big H present. And 50% will not have fur because there's no big H present. So that answers question number 1. Question 2, what is the genotype ratio? Genotype is referring to the different letter combinations we see. There are only 2 letter combinations here, big H, and little h. So the ratio is going to be 2 to 2, or we can say 1 to 1. Next, the question is asking for the phenotype ratio. Phenotype is referring to the characteristic traits that are visible, like does the cat have fur or does it have no fur? These two with the dominant alleles present will give fur, and these two with no dominant alleles present will give us no fur kittens. So the ratio here is 1:1. Just want to add that the genotype and phenotype ratio happen to be the same here, but keep in mind that they won't always be the same. So these are our answers here. Let's try another problem. This problem is from the official ATI TEAS study guide. I included a couple of these so that you can get a sense of what they're looking for and how they word their questions. Use a Punnett square to show the likely phenotypes of crossing two heterozygous tall pea plants. The allele for tall pea plant is dominant. That last sentence is a major key, and when you see it, you should start there. What I mean is assign a letter to the dominant trait. So here I will assign the letter capital T to equal tall and to represent dominant. Next, let's write down our possible genotypes. Capital T, capital T, capital T, and little t, little t. The given information tells us both the plants are heterozygous and tall. So the genotype we're looking for must be heterozygous and it must have the dominant allele. Big T is homozygous, big T is heterozygous, and little t is homozygous. The only genotype that fits our description is big T, little t. It is heterozygous and it codes for a tall plant. So we're going to take this genotype and put it in our Punnett square. Big T, little t represents both of our parents in this particular problem. So next, let's fill in our boxes with possible offspring. The question is asking for the likely phenotypes. Phenotypes is referring to the observable traits like tall or short. 3 of these squares will yield a phenotype of tall because they all have a big T. That one left over, little t, will yield a phenotype of short plant because it is 2 little t's. The answer given by ATI was this. Heterozygous crosses results in a 3:1 phenotype ratio. In other words, we have 3 tall plants to 1 short plant. If we cross big T, little t genotype with another big T, little t genotype plant, we're going to get a 3:1 phenotype ratio. This next question is also from the ATI TEAS study guide. It states, pea plant seeds are either yellow or green. Green seeds are dominant to yellow seeds. 2 pea plants that are heterozygous for seed color are crossed. What percent of their offspring will have green seeds? So the first thing you want to examine in the given information is what is given as dominant. Here it says green seeds are dominant. So we'll first assign a capital letter to represent green seeds. I'm using the letter S for seeds. Big S will represent green seeds, and by default, little s will represent yellow seeds. Let's look at the possible genotype variations. We can have big S, which is homozygous and will yield a green seed. We can have big S, which is heterozygous and will yield a green seed. And we can have little s, which is homozygous and it will yield a yellow seed. The given information says we have 2 pea plants that are heterozygous for seed color. That's what we're working with to find our genotype for both parents. It looks like only one genotype fulfills that description. Big S, little s is heterozygous. It doesn't tell us the color it yields, but it doesn't matter in this particular problem. So we're going to fill in this Punnett square appropriately. I'll give you some time to do that. Okay, so the question is asking what percent of their offspring will have green seeds. If the offspring is to have green seeds, a big S must be present. So these 3 out of 4 will come out with green seeds. The answer given by ATI was 75% will be green. Next, we're going to try a bit of a harder question. This problem is involving 2 traits, therefore we need to upgrade to a dihybrid square. Given the following information, how many seeds will be green and wrinkled? So color is one trait and the shape is another trait. Okay, so given information is right here. From this, we can see that round shape is dominant and yellow color is dominant. We're also given the genotype for each parent. The X in the middle just means that they're being crossed. It's a bit hard to tell big Y from little y here, so I'm just going to highlight big Y for the time being. The very first thing we're going to do is FOIL. FOIL stands for first, outer, inner, last, just like algebra. So first we get big R, big Y. Outer, we get big R, little y. Inner, we get little r, big Y. And last, we get little r, little y. Okay, so I'm going to let you try to FOIL the next one on your own. Awesome. So now we're going to use these genotype combinations and put them into our Punnett square. These are all for the first plant parent and we will put them up here. And these are all for the second plant and we will put them down here. By the way, it doesn't matter where you put parent 1 or parent 2. When you cross them, it's all going to be the same. So let's fill in a couple of these together. First, we have big R, big R, big Y, big Y. Next, we have big R, big R, big Y, little y. Next, we have big R, little r, big Y, big Y. And I'm just going to go ahead and give you a few minutes so that you can try and fill out the rest. Take your time because this part is really crucial to getting the correct answer. Okay, so I'm going to make some room here and push this stuff to the side. And I just want to remind you that R represents the shape of the seed. So if there's a big R present, it will give you a round seed. And if there is no big R present, then it's going to give you a wrinkled seed. Y represents trait number 2, the color of the seed. So if there's a big Y present, then the seed will be yellow. If there is no big Y present, the seed will be green. Okay, so the question is asking how many will be green and wrinkled? These 4 that I circled will be wrinkled, and these 4 will be green. Only one of these will be both wrinkled and green. So your answer is 1. Let's do a bonus question. How many of these seeds will be yellow and round? I'll give you some time to think about it. These 12 will be yellow, and these 12 will be round. So how many are both yellow and round? 9. Awesome. 9 will be both yellow and round. Okay, so just remember when you are presented with a question involving two traits, you need to use a dihybrid square with 16 boxes, and remember to foil first. Okay, so we're done with the fun stuff. We know how to fill in a Punnett square and all. There are a few facts you need to know as well as exceptions you need to know. So let's go ahead and review those really quick. Gregor Mendel is the father of genetics, and he studied genetics using pea plants. He found how genes are transferred from parents to their offspring. So you must be familiar with Mendel's law of heredity. There are 3 in total. The first is law of dominance. It states that dominant alleles mask recessive alleles, thus making the dominant alleles express. So you should understand this first law fairly well because this is exactly what we've been going through in every single practice problem. The second is law of segregation. Alleles are separated in a way that each gamete carries only one allele for each gene. This one is a bit hard to visualize, but the concept is super simple once you're able to get it, once you're able to visualize it. So if you look back at our previous problem, notice when we broke down the genotypes for the parents, in other words, we foiled them, we got these gametes that each had either a small r or a big R, but not both. And they each had a small y or a big Y, but not both. This is what the law of segregation is. It means you won't have both alleles for one gene in one gamete. Instead, the 2 alleles split into 2 different gametes. Okay, so the third law is law of independent assortment. Alleles do not influence each other when they segregate into gametes, and thus each combination of different alleles is equally likely. Alrighty, so let's lastly go over a few exceptions to Mendel's law. These are also referred to as non-Mendelian traits. First, incomplete dominance. This is when the dominant allele is not fully expressed. In this situation, there is a blend. For example, in Mendel's law, if we have a dominant allele and a recessive allele together, dominant will automatically mask the recessive, right? And the dominant trait will completely show. So here, the dominant trait is blue. But in incomplete dominance, the dominant is only partially expressed, which means there is a blending of both the dominant color and the recessive. So for incomplete dominance, think of a blend. In codominance, both alleles are fully expressed. Emphasize on fully. So for example, we have a yellow fish and a blue fish. We end with a fish that has both fully expressed alleles, both fully expressed colors. The fish has both yellow and blue. It's not a blend. So if this was incomplete dominance, then the offspring would be a mix or blend of whatever blue and yellow make, right? Okay, so lastly, we have polygenic inheritance. This is when many genes influence one trait. For example, height is influenced by many genes. So here, AA is one gene, BB is another gene, CC is another gene, and DD is another gene. They all work together to create height. Alrighty, everyone, that's it for this video. Thanks for sticking around. I hope you learned something new, and until next time.